Now on to today's RettEd topic, MECP2 and Rett syndrome. How do your child's genes fit into gene therapies? With Dr. Eric Marsh, Medical Director, and Holly Dubs, Licensed Certified Genetic Counselor at IRSF's Center of Excellence at Children's Hospital of Philadelphia. We've invited you to this RettEd session for a comprehensive overview of genetic therapeutic advancements for families and caregivers. Dr. Marsh and Holly will explore the foundational principles of the genetics of Rett syndrome, including a review of the basic biology processes like transcription and translation that underlie genetic-based treatments. We'll delve into cutting-edge therapeutic genetic approaches such as RNA therapeutics, gene therapy, and gene editing, and they'll cover delivery methods and potential side effects. You'll also learn how outcomes are monitored and optimized. Thanks for submitting your questions in advance, and please submit additional questions that come up throughout the presentation, and we'll answer and address these during our QA at the end of their presentation. Okay, so with that, I now invite Dr. Marsh and Holly to take the virtual stage and help us better understand MECP2 genetics and gene therapies. Dr. Marsh, Holly, you ready to screen share?
All right.
Thank you so much.
All right. Well, welcome everyone. Good morning, good afternoon, good evening, depending on where you're located. And we're so excited to have people joining us from all around the globe today. What our plan for today's talk will be is to first lead off with an overview of the basic genetic processes. This is not meant to turn anyone into a biologist, but want to help kind of bring you all up to speed and make sure we're on the same page when we start to talk about the area where most of you are likely interested in terms of some of the genomic medicine techniques. We'll incorporate a review of Rett syndrome genetics into that talk, and then we'll transition to talking about some of the emerging clinical work and preclinical work that's being done to explore genomic medicine. So just to start off, this is information that many of you may be familiar with, but we know that Rett syndrome was originally described in the 1960s by Dr. Andreas Rett when he noticed that several patients that he was following had very similar clinical phenotypes, which are the symptoms that many of you are all very familiar with relating to your loved ones with Rett syndrome. There was a more formal description and a clinical phenotype which was described in 1983. And really, in terms of the genetic landscape, we started making a lot of progress in that field in 1999 when it was first found that mutations within a gene called methyl CpG binding protein 2, or MECP2, located on the X chromosome, were found to be the cause of Rett syndrome. We know that Rett syndrome affects approximately 1 in 10,000 live births. And in 2002, there was another formalization of the clinical criteria for Rett syndrome that was published. And these, again, are kind of many of those features that you are familiar with. And at that time, sort of before we had good access to genetic testing, people who had those clinical symptoms and who met those clinical criteria were given a clinical diagnosis of Rett syndrome. Once we started doing genetic testing in those individuals, we found that in somewhere between 95 to 97% of people that met those criteria, we were able to identify a pathogenic variant in the MECP2 gene. And we had a follow-up of a revision of those diagnostic criteria published in 2010 by Dr. Newell and his colleagues. And those are really still the clinical guidelines that we use today. So the clinical criteria that were published in 2010 are still the clinical criteria that we use today to discuss whether or not someone's symptoms fit with a Rett syndrome diagnosis. When we start to think about gene therapy and therapeutics and treatments for Rett syndrome, the field really shifted in 2007 when we discovered that symptoms of Rett syndrome in a mouse model were able to be reversed. So prior to that, given the regression that many individuals with Rett syndrome experienced, it was uncertain whether or not, if at some later point in time, one was to intervene in some sort of way, would we be able to reverse those symptoms, or were those changes permanent? And with some of the experiments that were done in 2007 by many of our colleagues, we were able to discover that those symptoms could potentially be reversed even after symptoms were already present. And that's really paved the way for much of the research that's been done over the last decade and a half, looking into ways that we can potentially bring these types of therapies to people, which is what we really are all hoping to see. So when we think about the genetics of Rett syndrome, we know that within the MECP2 gene, over 1,000 different variants have been described. So there are many different ways the gene can be altered that can lead to Rett syndrome. However, we know that about 8 common variants represent approximately 50% of all the genetic changes in MECP2. And I have some of those most common variants listed here. Some of you may recognize them as genetic changes that might be present in your loved one. And where this becomes sort of a useful piece of information when we think about therapeutics is we will hear later from Dr. Marsh that there are some therapies that might have kind of a broader impact and be relevant for broader genetic landscapes, and there are others that might be more specific. But by knowing that many people have the same exact genetic change, even if we start to talk about very specific targeted therapies, we know that there will still be a large percentage, maybe not everyone, but a very large percentage of individuals with Rett syndrome who would still benefit. Transitioning a little bit more to some of the nuances of DNA and some of those biologic processes, when we think about the nomenclature or how a genetic variant is written out, we typically see 2 different terms referenced, one starting with the letter C and one starting with the letter P. And those represent the same genetic difference, one being at the level of the DNA and one representing the level of the protein. What do I mean by that? So let's talk a little bit more about some of the underlying genetic biology. So for those of you who might remember back to some high school biology courses, our DNA is encoded by a 3-letter code that is A, C, T, and G. And within the DNA structure, it's a double helix with those letters pairing up with one another. T links with A and G links with C. And each 3-letter fragment of DNA encodes for a particular amino acid. Those amino acids ultimately go on to become the building blocks of proteins. And so the chart and table here just shows how those different DNA base pair combinations can lead to different codes for different amino acids. You will see the letter U is represented in that big table rather than a T, and that's because a U is substituted in the molecule called RNA, that it serves as sort of the messenger to translate from DNA to protein. So some of those terms that I was just using, I wanted to explain in a little bit more detail to you. So DNA is basically the equivalent of a recipe book that tells our cells how to make things in our bodies. They basically encode all of the recipes or the sort of underlying code that then our body takes to interpret and takes to ultimately produce those proteins. RNA is basically the messenger or the instructions that would go to a chef to know how to cook or to make things in the cells, kind of using that cooking analogy. And then proteins are ultimately sort of what the chef makes. They are the substances or the molecules that do everything within our cells. Everything within our body is composed of proteins, and those proteins are made of building blocks called amino acids. The process to go from DNA to an RNA messenger is called transcription. And the process to go from that messenger RNA to a protein is called translation. So those are all important steps of the DNA process that can either A, be disrupted when a genetic variant is present, or can be areas where we can think about potentially intervening in terms of therapeutics. When we think of genes and mutations and genetic conditions, we typically have our normal gene, which is basically kind of the standard code that everyone should have. And that goes on to produce a normal protein that is able to work properly. It's able to do its job the way that it is supposed to. When we start to think about genetic conditions, there is at some level a difference in the way that that gene is being coded. We call that a mutation. And depending on the exact nature of what that mutation is, it can result in several different effects in the protein. So in some instances, we have a genetic change or a mutation that results in no protein being made. In other instances, it results in a protein that's abnormal, and often that abnormal protein is no longer able to do its job properly. Taking that a step further, we know that there are other sort of areas of our DNA that play an important role, other important areas of our DNA they can have changes in that can affect this whole machinery. So one of the important areas of our DNA is something called a promoter. Basically, a promoter is like the signal that cues up the genetic machinery to get to the starting line, so to speak. So the proteins in our cells kind of recognize that promoter and use that to kind of locate that starting line of DNA to help transcribe those genes. At the end of the gene, there's a stop signal and a termination codon that tells the transcription machinery to stop. The body then goes through a process of what we call transcription, where it takes an mRNA instruction or messenger that was made from that DNA and uses that mRNA to make a protein. What we know is that our body is trying to fit a lot of information into a very, ultimately very small bit of DNA. And so we know that our body has found ways to kind of make that machinery more efficient. One of that is through a process that we call splicing. And that is a process by which the same gene and the same sort of code of DNA can be used to make multiple different proteins. So when we think about our DNA sort of at the gene level, we have building blocks called exons and regions called introns. So you'll see here on the slide, the blue, green, red, light blue, and yellow blocks are numbered exon 1, 2, 3, 4, and 5. Those are the regions of the DNA that specifically are coding for the protein products that are made from that piece of DNA. The DNA regions sort of in between those exons are called introns, and they ultimately get spliced out. They ultimately get removed from the final mRNA and protein product that goes on to make a protein. And you can see that depending on how we put those exons together, depending on how we match those building blocks up, it can result in slightly different proteins. And so again, this is a way that our body can use one strand of DNA, one gene to make multiple different protein products. And that process is called alternative splicing. At the MECP2 level, or thinking about sort of genetic variants that cause Rett syndrome, I think many of you may ask, what does my daughter's or my loved one's genetic change mean? We have several different variant types, and those can impact the protein in different ways. So the first type of genetic variant we can have is something called a missense variant. And that basically results in one single amino acid being replaced by another in the protein. And we kind of think of this as a genetic misspelling. Within the MECP2 gene, some common examples of common missense variants are T158M, R133C, R306C. And basically this just means that the first amino acid is replaced by a different amino acid. The next kind of genetic variant that I wanted to mention is something called a nonsense variant. Basically, what a nonsense variant is that the DNA change results in a stop signal in the protein. So instead of stopping at the end where it's supposed to, that stop signal occurs much earlier in the protein. And that typically results in the body sort of recognizing, hey, this protein's not made the way it should be. Let's just degrade it. Let's break it down. And no protein is made. And some common examples of that in terms of MECP2 changes are R168X or R270X. So basically that X at the end of the variant name indicates that that's a nonsense variant or a nonsense change. Another common type of genetic variant that we can see is something called a frame shift variant. If you remember, as I mentioned a little bit earlier, our DNA is read in those 3-letter codes, and each 3 letters encodes for an amino acid. So if you either add or take away pieces of DNA that are not in a multiple of 3, what happens is that everything that comes after that winds up getting shifted over. And that means that each 3 letters is now typically coding for a different amino acid. And that ultimately tends to result in a premature stop at some point before it gets to the very end. And so some common examples of this are variants like 806 del G, 710 DUP-G, and then larger differences like 1157 to 1197 del. And basically that means that all the DNA from spot 1157 to spot 1197 is all missing. Everything that comes after that has been shifted over and ultimately will result in a stop. And those are easier to sort of recognize at the DNA level nomenclature instead of the protein level nomenclature. We can also see intronic variants. These are less common in MECP2, but they certainly happen. And basically what these are differences that occur in the DNA sequences in between the exons. So they are differences that are not affecting sort of the coding parts of the DNA directly. However, they occur in the sequences in between that ultimately influence or affect the way that the DNA is processed. And so in those instances, the exons might not be put together the way that they're supposed to be to make the protein properly. And we can usually recognize those types of variants because they typically include a minus or a plus sign in their nomenclature. And then the last type of difference I wanted to mention are exonic deletions. And so these are basically larger pieces of DNA that are missing. I mentioned earlier within frameshift variants, sort of small numbers of DNA base pairs that are missing. These are usually sort of larger chunks of DNA that are missing that can include, for example, multiple exons or multiple of those building blocks of the MECP2 gene. So something that we see commonly in individuals with Rett syndrome is that they're missing exon 3 and 4 of the MECP2 gene. And how this becomes relevant is because it can begin to impact the way that we think about potential targeted treatment strategies. So historically, most medications and most drugs that people take are things that are not impacting the DNA directly. They're often molecules that will impact downstream targets of whatever sort of the disrupted pathway is. So when we think about MECP2 specifically, we know that MECP2 has a lot of roles or a lot of jobs that it does within the body, and some of them are listed here. One of them being that it impacts growth factor signaling, and that's really the pathway that's targeted by trofinetide or DAYBUE. We know that in individuals with Rett syndrome, that pathway is disrupted. DAYBUE aims to sort of regulate that pathway as a treatment strategy. And so that's kind of really where the focus of traditional drug and traditional small molecule therapies has been focused. When we start to think about genomic and genetic testing strategies, we have a lot of different ways and a lot of different aspects where we can think about potentially targeting interventions to ultimately impact or alter the way that the gene is working. And so now I'm going to turn it over to Dr. Marsh to talk to you a little bit more about specific genomic type therapies.
Thank you, Holly, for that wonderful setup. So now we're going to transition from the backbone and the basis of how we can manipulate the genome such that we can potentially impact symptoms and the phenotype and the picture that a child with Rett syndrome or any other genetic disease can have. So when we talk about genomic medicine, we talk broadly about using the person's DNA to help them stay or get healthy. A lot of people talk about individualized medicine, personalized medicine, or precision medicine. And these are all novel ways to use someone's genetics, or the other option for personalized medicine is their specific environmental factors that make a more precise approach for the prevention, diagnosis, or treatment of disease. And today we're going to focus on the genetic side of this. Next slide. So as Holly said, you can go from your gene of interest to a potential therapeutic. And there's multiple ways to do this. You can target the RNA that gets produced from the DNA. You could target the DNA itself, or you can replace the DNA, provide new DNA. With the ultimate effect of all of those is to have a healthy or a appropriate copy of the MECP2 protein in cells so that the cells can act normally and function properly. So there's many ways to do this, and this slide just gives kind of an outline of all the different ways that one can do this. Next slide. So we're going to break this down now into the kind of the three major buckets that exist for these genetic-based therapies. And we're going to start with RNA therapeutics, and then move into gene therapy, and then DNA editing at the end. RNA therapeutics are not in the clinic yet for Rett syndrome, but there are numerous companies who are working on this, so this will likely get to the clinic sooner rather than later. And RNA therapeutics uses small pieces of DNA or modified RNA to target those RNAs, the messenger RNA or the transfer RNAs, in order to change the amount of protein and which protein is expressed, so that you can have normal protein within the cell. And so you can do that in multiple different levels. So as Holly already explained, you go from the gene, which is the top, I don't know if you guys can see the pointer when I do this, but which is the top row of this image, and that's the DNA, that's the gene. And then you get transcription of an RNA, which is that middle one. And so you could target aspects of that transcript in order to make it go away or to persist longer, such that you have more of the RNA being produced. And then you go on to making a mature RNA, and that requires a number of different steps that could be targeted by these small pieces of DNA or RNA. And then lastly, that mature RNA becomes protein. And that step of going from mature RNA to protein requires little pieces of RNA called transfer RNAs, and you can target those such that you can make sure that those stop codons that Holly mentioned before, those nonsense mutations, can be read through so that you have a normal, healthy, wild-type copy of MECP2 in cells. Next slide. So there are a variety of different flavors of these RNA therapeutics. And so terms you'll hear about these RNA therapeutics are ASOs, or antisense oligonucleotides. And these are generally pieces of RNA that have been modified, or DNA. They're a single strand. So we know DNA is double-stranded typically. These are single-stranded molecules that can block or alter RNA processing. You can have RNAis, or interference RNAs, and these are little double-stranded pieces of RNA that bind to inhibit or alter the messenger RNA. And then you can have siRNAs, or small interfering RNAs, and these are really small. They're 21 little bases of a double-stranded RNA that can interfere with translation or that transcription step by impacting those transfer RNAs. And all of these different approaches to modifying the process from DNA to protein have been used on a research basis, and all of them are moving towards clinic. And one of the things I've said a couple times now is modified RNA. And the reason why you have to modify RNA in order to use it as a therapeutic is that the backbone of RNA by itself is not very stable, so that if you were to give someone a piece of RNA, your body would just eat it up within seconds. So it doesn't have stability, whereas DNA is very stable. You can give someone a piece of DNA, and that DNA will remain intact for a long period of time. The best example of this is that dinosaur DNA still sits from thousands and thousands of years ago, but there's no dinosaur RNA because it disappears because of enzymes that chop up RNA very easily. So because of that, what the companies and what scientists have thought about is that you can modify the chemistry of the RNA such that those enzymes that break up RNA no longer work, and now RNA can be used as a therapeutic. The example of this that we all know really well is the COVID vaccine. So the COVID vaccine is a messenger RNA vaccine. It gives a piece of RNA that goes into your muscle cells. Your muscle cells take up that RNA. It gives the instructions to make a part of the COVID protein, the COVID vaccine capsid, which then goes onto your muscle cell, and then your body makes an immune response to the COVID vaccine. And that COVID mRNA is a modified RNA that's stable and can be given to people. Next slide. So how does one get RNA into the body? And this is also true with DNA therapeutics. So I'm mentioning it here for RNA. I think I come back to it for DNA therapeutics, but there's multiple different ways to get it into the body. And so these are not something right now that you could take by mouth. The simplest way to get these into the body would be by IV, which is the image on the left of the screen. And that is going into a vein. And if you go into a vein, the RNA therapeutics have to cross the blood-brain barrier in order to get into the brain. But it's the easiest way to do it. The problem is many of these RNA or DNA-based therapeutics can have trouble getting across the blood-brain barrier into the brain. So right now, for neurologic disease, IV is not the route of administration. The next route, and I'm going to skip to the image on the right, which is via LP or lumbar puncture. And this is where you go into the CSF, the cerebral spinal fluid that sits around the brain and goes everywhere around your brain. And at the bottom of your spinal cord, below the level of your spinal cord, you have a little reservoir of CSF. And so you could put a needle between the bones into that reservoir of CSF. You could do it very safely. This is something that's done, thousands of times a day for women who are getting an epidural, for people who have potential meningitis, et cetera, and it's done safely. And so you can go into that space, the bottom of the spinal cord, and you can inject the therapeutic, and it will then diffuse all around the brain and hopefully hit everywhere in the brain. The limitation of this lumbar puncture, or the other term for this is intrathecal IT approach, is that it has to go down, it has to go upstream, in that gravity, everything else pulls the fluid down to towards your legs. So the therapeutic has to go upstream. But as you know, from like when you put a drop of dye in water, things diffuse throughout the solution. So this therapy will diffuse throughout the brain and get most places in the brain. The other ways to get, if you want to get to the cortex, to the main thinking part of the brain the most, is to go directly towards that area. And there's two approaches that. One is this ICM, which is intracisterna magna, and that is using the space that sits below the cerebellum and injecting the therapy into there. And the last is intracerebral ventricular, which is going into the large fluid-filled spaces that sit in the middle of the brain. ICV requires a surgical procedure because you have to put a hole in the skull, and then you have to go through the brain into this fluid-filled space. So it's the most invasive, but it's also something that can be done very, very safely by any neurosurgeon. So all four of these approaches have their strengths and their weaknesses, and there's different reasons to use one versus the other, and a lot of it depends on the therapeutic that you're giving as well. Other thing about RNA therapeutics is that while these RNAs are stable, they're not super stable, so that they do have a half-life. They do disappear with time, so that you do need to do repeated delivery. And the half-life of each of these is going to depend on the chemistry of them, and it can be the half-life can be from weeks to months. So for certain of these therapies, you're going to have to dose it, say, every 2 months or every 3 months or every 4 months, depending on the chemistry of the molecule. And then the last thing is that you can actually, everything I've talked about up to this point has been delivering this RNA therapeutic by itself, but you can actually put the RNA therapeutic in a carrier that allows a cell to take it up and produce that siRNA or ASO by itself, which then acts directly into the cells. And that is similar to what we do in gene therapy, but as opposed to providing the gene back, it's providing a piece of RNA that then acts on the messenger RNA or the transfer RNAs or some step in the process that Holly went through. Next slide. So an example for this that you might have heard about is that there are actually 2 clinical trials going on using an antisense oligonucleotide in Angelman syndrome. Angelman syndrome is one of the other well-studied, historically known neurodevelopmental disorders. Its genetics is a little bit more complicated than MECP2 because it turns out that there's a multiple different genetic mechanisms, but what these ASOs do is they inhibit a RNA that is normally produced by the cell that prevents expression of this gene UBE3A. So people who have Angelman's aren't expressing UBE3A, so you can give an ASO to inhibit an RNA that inhibits the UBE3A. I know that's very complicated, but they're showing very positive things in the 2 trials that have moved forward for Angelman syndrome, and they're about to start a phase 3 trial. And a phase 3 trial is a trial where you prove that the drug works after you've already shown that it's safe in phase 1 and phase 2 trials. So that's a great example of another neurodevelopmental disorder. An ASO is being used. And then the other example would be Spinraza. This is for spinal muscular atrophy, or Lou Gehrig's disease in babies. And that has also been shown to work and is already FDA approved. Next slide. So there are a number of RNA therapeutics that are already on the market. And this is a listing of them as per, I think, 2019. So it's actually even more out there now. And you see that they could be used to target both brain conditions, such as spinal muscular atrophy, or Batten's disease, which is Millicin, though that's not actually FDA approved per se. But it also could be used for treating muscle disease like Duchenne's muscular dystrophy, but also blood disorders and liver disorders like acute hepatic porphyria listed down below. So these can be used for any disease, and we're at the cusp of seeing these move forward for neurodevelopmental disorders like Rett syndrome. Next slide. So when you talk about therapies, you do have to talk about side effects. None of these, no drug is without potential side effects. And so there are potential side effects. And one of the things that we're learning from these RNA-based therapies, that there are certain side effects that appear to, almost regardless of the chemistry or regardless of the target that is being targeted for the different conditions. And one of the things that people have seen, they saw in the Angelman study, was that kids became transiently weak. They got leg weakness, but that resolved. They all ended up going back to walking. That you can see suggestion of inflammation because you have fluid protein in your spinal fluid gets higher than it's supposed to be. Right now, we're not sure the exact impact of that increased spinal fluid protein, but there have been some cases where it is possible that that increased protein has led to brain swelling and hydrocephalus. We know that these RNA therapeutics can impact the kidneys. They can kind of clog up the kidneys, so you have to be very careful in treating people who have kidney issues with it. And then there are a variety of systemic symptoms that can occur. These are symptoms that can affect your blood sugar, your blood pressure, and they're all listed there. But as the previous slide showed, that a lot of these have moved forward and been approved, we're aware of these side effects. So for any clinical trial that moves forward with an RNA therapeutic, the investigators and the sites would be very aware of these side effects, and these would be things you'd be looking out for as you do the clinical trial. Next slide. Okay, so now we're going to shift to gene therapy, and this is what's actively going on right now for Rett syndrome. And this is modification of the cell's genes to produce a therapeutic effect. It is giving back a copy of the gene so that the cell has the gene that it needs to function properly. And there's two ways to do this. Gene replacement, which is what the current Rett syndrome therapies are, and gene editing, which we'll get into at the end of this talk. Next slide, please. Oh, sorry, go back one, Holly, sorry about that. And so the schematic on the right shows what you do in this, is that you take a piece of modified DNA, and that's shown in the upper left-hand corner there, and your gene of interest sits between viral pieces of DNA, and you package that in a virus. And the virus that people use now is adeno-associated virus. And the viruses infect cells. So the virus infects the cells in the brain, and it drops off the DNA. And the cell then allows that DNA to stand there, and it recognizes that DNA such that it produces RNA and then produces the protein that you need. Next slide. And many people think about this as a Trojan horse, right? You are tricking the cell to allow a piece of DNA into it that it wouldn't necessarily want. And the two images on the right are 3D reconstructions of what these viruses look like, and shown in all the different proteins in different colors. So it's a beautiful, colorful image of the virus. And then on the lower right shows these little stalks that stick out in green, and that's what actually helps the virus bind to cells. And researchers are spending a lot of time trying to figure out ways to get these viruses to bind to specific types of neurons, or neurons in general, really efficiently, so that you can get the gene that you want into all the cells that you want. There are a number of different viruses that can do this. One of the first ones people used was adenovirus, but unfortunately, we are exposed to a lot of adenovirus. It's the common cold virus, so that your body often makes an immune response to it. So it's not something that's being used therapeutically in a person, though it is being used, can be used therapeutically in cell culture, then when you put those cells back in a person. Lentivirus is also another virus type that we do get exposed to. So it's also something that your body would make an immune response to, but it's also being used for things like CAR-T therapy or ex vivo out-of-the-body gene therapy. An example of out-of-the-body gene therapy is an example for sickle cell disease, where you can replace the gene in someone's blood cells or their blood stem cells, and then you can put those stem cells back in the person, and then the person makes normal hemoglobin, so they no longer have sickle cell disease. The other two ways to get these DNA pieces into cells are using nanoparticles, and this is coating the DNA with gold or with other chemicals that allow the DNA to get into a cell, and then the cell recognizes it and takes it up, or putting it in little lipid droplets, which is very similar to a virus, except without the protein. But you can do that, and the cells will take up these lipid particles, and ultimately to replace the gene of interest, to replace the gene that is damaged because of the mutation with a healthy copy of the gene, so that you produce normal amounts of protein. Next slide. So the steps, I think I kind of went over this, but I'll just go it over again quickly to make sure we all understand it, is that virus goes in to the cell. It then gets picked up by this stalk protein that binds to a receptor on the cell surface. It gets internalized into the cell. The virus gets dumped out. The virus opens up, which is the uncoating step. It then releases its DNA molecule, which then turns into a little circle, which is called an episome. And then that episome is recognized by the cell's normal machinery to produce RNA. So it produces RNA. The RNA goes out of the nucleus into the cytoplasm, where the machinery that turns the RNA to protein happens, and then you get normal protein in the cell. So this is a way to bypass the mutation to produce normal MECP2 protein. And that's what the labs, the companies that are moving forward with gene therapy in Rett syndrome are doing. Next slide. So there are multiple different ways to do this, and not going to go into super details here, but when you make this little piece of DNA that goes into a virus, the virus only can hold a certain amount of cargo. And in doing this, you need to keep some of the viral sequence there so that it can be taken up by the virus, and so that it can form this episome, or this circular piece of DNA within the cell. So those are these ITRs that are shown down in the lower left-hand image, and those are called inverted terminal repeats. And so this is what needs to be on there. And then you need a promoter. You need something to tell the cell to start producing the transgene. And then you need a tail of it so the cell recognizes it as normal human mRNA. And so when you do this, you can either do this as a single-stranded molecule, or you can do it kind of as a double-stranded molecule. And there's strengths and weaknesses in both. And indeed, in Rett gene therapy, one of the companies uses a single-stranded molecule so that they could put in the entire MECP2 protein. And the other does this double-stranded or self-complementary AAV, so that they need to use a mini version of MECP2, which has been shown in labs to be effective in producing protein that functions properly within the cell. And the reason why you'd use one over the other, there's strengths and weaknesses of both. The single-stranded version is thought to take longer to produce the protein, and it does not do it as efficiently. So you need more virus, you need to get more of it in there than in the self-complementary. But the downside of the self-complementary is that the entire MECP2 protein doesn't fit. So you have to use this kind of smaller version of the MECP2 protein. But as I said, labs have shown that that smaller version has been effective in mice and in cells to perform the functions of the normal MECP2 protein. Next slide. So as I said, for RNA therapies, there's different ways to get this into the brain. So you can do it by IV, intrathecal, cisterna magna, intracerebroventricular, or you can direct directly into the brain. And there's actually an FDA-approved gene therapy for a pediatric onset movement disorder where they do exactly that. They inject it into the basal ganglia of the kids because that's where the main genetic mutation is, and that's been shown to be very effective. You need large amounts of virus. So if you guys remember from high school math, scientific notation, that when we talk about these viruses, we talk about it in the number of copies in scientific notation. So this is 10 times or 1 times 10 to the 13th copy. So that is times 10 to the 6th is 1,000,000 copies. So this is 1,000,000 or 10 million copies. So it's an awful large amount of virus, but you need that much to get it into all the cells in the brain. The gene therapy approaches produce prolonged expression. So this is a one-time dosing. You only need to give this once. And from what we know, both in mice and in non-human primates, and so far in kids with SMA, once this gets into a cell, it seems to continue to express for a very long time. And there's no evidence in kids who've gotten the SMA gene therapy 10 years ago now for any change in the expression of the gene. So we think that it continues to express for the life of that cell. And since brain cells stay alive for the entire life of the person, that we think it would remain expressed for the life of the person, meaning that it is a one-time dosing. But because of this, some practical issues. If you give it a virus, you can't have exposure to this AAV virus. And while it's not common for people to have exposure to these AAV viruses, there is some exposure out in the community. So before you do gene therapy, you get tested for the presence of the AAV. And if you have too many antibodies against that AAV, you might not be able to move forward with gene therapy. It's something that many people are thinking about as ways to still allow people to move forward. But at least in the trials, if you have too high of antibody levels against the AAV, that is an exclusion criteria. Because you've now exposed someone to a lot of this virus, the risk of redosing is that the person's going to have a tremendous immune response to it. And that immune response can be deadly, as we've seen unfortunately, very unfortunately, in the neurogene case that you're all likely aware of. And because of this potential immune response, people get immunosuppressed. So you shut down the immune system so you can put in this huge amount of virus without the body waking up to it and attacking the virus so that it can get into cells. So that requires immunosuppression. This is something that scientists are still trying to figure out, and researchers are still trying to figure out about how much immunosuppression you need and for how long. And it's something that, as these trials move forward, this is the type of thing that it's going to evolve as we learn more and more about what is the best way to do this. Next slide. So, like with RNA therapeutics, there are safety issues that we know about gene therapy, and some of this is based upon the AAV. Some of this is based upon putting in this extra pieces of DNA, and there are questions about it causing DNA damage. This has been shown in humans, but it's unknown if this happens in humans. The inflammation that happens, the immune response that happens, can cause injury to the liver. And there's some question of whether this is direct from the virus or only immune, but both of them can happen. And so you have to be really careful in preventing this from happening. And corticosteroids can work for that. You get a bleeding problem where you get damage to the small little capillaries and endothelial cells, which then cause your immune system to respond, and that can be dangerous. And so you want to prevent that from happening. These cells, this virus goes really well into your dorsal root ganglia. These are little clusters of neurons that sit outside your spinal cord. And the question is that going to cause those cells to be damaged, and what's the long-term effect of that? And so far for spinal muscular atrophy, we haven't seen any issues, but we know that this happens in non-human primates. Then there's the question of that there's some immune response that's happening within the brain, and does that cause injury? And right now, we don't know really why that's happening, and we don't know how to prevent it. And we also don't know if it's due to the actual delivery or if it's due to the virus or the gene. And it doesn't seem to right now in spinal muscular atrophy or the other gene therapies that have moved forward to have a significant clinical impact on anybody at this point in time. So we all believe that it's safe to move forward, even though we can see little things happening. Next slide. And so, as I mentioned, you have to do immunosuppression. So these are the types of drugs that people have been using to try to modulate the immune system so that you can get the virus into the cells without causing a major immune response that can be dangerous to the individual. And so the classic ones that people are using first are steroids, rituximab, and things like rapamycin, which are the top ones listed up there. Next slide, please. So, as you know, there's 2 active therapy trials in Rett syndrome by Neurogene and Taysha. And on the slide on the right is a recent take of gene therapy trials going on for multiple different disorders. And you see there's 67 trials going on for neurologic disorders, many for eye disorders, heart disorders, metabolic disorders, etc. So we are learning an awful lot about gene therapy as a platform, as the virus. And so learnings from all of these different trials will help us improve how we do the active trials in Rett syndrome. And individuals in these companies are both very actively involved in knowing what's going on in all these diseases to improve their trials moving forward. Next slide. And so there have been a number of gene therapies that have been approved. Some of these are given directly into the body. Some of these are actually given to blood cells outside the body or given to immune cells outside the body to do what's called CAR-T therapy, which are modified T cells that attack cancer and have been shown to be a very effective cancer therapeutic. Next slide. I'm running out of time, but I think I only have 2 slides left here, is gene editing. So this is something you've all probably heard about. It's called CRISPR. It uses a protein called a Cas9, which is a bacterial protein that can go in, find mistakes in the DNA, and then correct those mistakes. So what you do is you put in a copy of this Cas9 protein, you put in a guide to direct the Cas9 protein to the mutation that you want to correct, and then the DNA, that Cas9 protein, cuts the DNA where you've directed it to it, and then the body proofreads it and puts back in the healthy wild type version of the base pair, so that you go from a mutation to normal. And so those 4 steps are shown there in cartoons. And it's been shown to be very effective in cell culture and in mice. And people have now done it out of the body for hemophilia and for sickle cell disease. And they're starting to do therapies in the liver where you direct it directly at the liver. Next slide. So part of the problems with gene editing is that while you give the process this little piece of DNA to direct it where you want to go, it doesn't always go to where you want it to go. So it can go elsewhere within your DNA and cause breaks and cause mutations. And so the first generation of these, which came out over a decade ago, were very dirty and would cause many, many mutations. But scientists have figured out ways to change that Cas9 protein such that it's much cleaner and it really only goes to where it's supposed to go. But it still hasn't gone to the point where it only goes to where it's supposed to go. So there's a question of, are you going to induce damage that will cause mutations? Now, a lot of the DNA is probably tolerant of mutations, so it might not be a big deal, but it's something that scientists have to be really careful and researchers have to be really careful about as they move forward. The second thing is that these Cas9 proteins are bacterial proteins, and our bodies don't like bacteria. If a bacteria gets into your body has a big immune response to it to fight it off. And one of the things that are most evolved in humans, besides our intellect, our speech, and our ability to manipulate things with our thumbs, is our immune system. The human immune system is rather unique in how strong and effective it is. So there's a concern about putting these Cas9 proteins, these base editing proteins in, and ultimately cause problems if you do that. So that's why the therapies that have moved forward have been outside the body, because you can wash away the Cas9 before you put it back in, or going directly into the liver, where you can do a one-time dosing and the liver cells will get rid of it so that you don't have to worry about that. So those are the kind of the system-wide problems that we have with gene editing, but there's lots of companies and a lot of scientists and a lot of academic institutions around the world who are trying to solve these problems, because the potential for this is tremendous. Next slide. And so just to end, as I said, there actually now has been 2 approvals for ex vivo therapies. So these are outside of the bodies. And this is sickle cell disease and beta thalassemia. And they've been approved and are very expensive. And there's people learning how to use them. But it really is just the cutting edge of what we can do in these genomic medicine approaches. Next slide. And with that, we'll take questions.
Great. Thank you, Dr. Marsh. Thank you, Holly. Okay, tall order to ask you to explain the genetics of Rett syndrome and potential gene and RNA therapies in 1 hour. And you did an excellent job. Thank you. Each of those sessions could take separately a couple of days to really understand it. And so we appreciate your speed and clarity. We do have a few questions that people submitted. And if we run out of time today, we'll certainly take those questions offline and send them out to the community. Okay. Holly, if you'd like to come back on camera and we can take slides down, let's go into just a few minutes of QA. So we did have a question about, can gene therapy make things worse?
So I guess there's two ways to think about that. So one is the side effects of gene therapy that we've talked about, and unfortunately seen in the Neurogene trial, right? That's a case where it really made things about as worse as it can get. So there's that aspect of it. And that's things that we hope we can manage and prevent as we move forward with this. The second part of that would be, could the gene therapy produce too much of the protein or do something else that would be problematic? And for Rett syndrome, which we kind of can think of like a Goldilocks protein, in that you can't have too little of it because that's Rett syndrome, and you can't have too much of it. That's MECP2 duplication syndrome. So you need the right amount of it. And both the companies that are doing this and anyone who thinks about Rett syndrome thinks about that a lot, on how to make sure that you only have the right amount of MECP2. So both these companies actually have little molecular tricks that they've put in their gene therapy to prevent having too much MECP2. But yeah, that is a possibility, that you're having too much MECP2, or it's also a possibility that in rare circumstances that the one protein interacts with the other protein and creates a problem. But we don't think that's going to happen because that hasn't happened in the mouse. So the big issue is too much or too little. But hopefully the mechanisms and the technology that people put into this will prevent that from happening.
Great, thank you. And that's why we do clinical trials, right?
Exactly.
Okay, so next question is, thanks for explaining how mRNA can degrade over time and that gene therapy typically is a much more stable intervention. The question is, can it be reversed or removed once given, or bring things back to the original state?
So unfortunately, no. So once this goes in, it's not like a tattoo, which now we can get rid of. Right now, once this goes in, it's in for life. We have no ways of going in and degrading that piece of DNA that's sitting in those cells. So it's permanent. So once you get it, you get it for life. Oh, you're muted, Paige.
Thanks, sorry. We also had a number of families ask about impact of DAYBUE, and if taking DAYBUE will make you eligible or ineligible for gene therapy trials or any approved gene therapies.
So there's two things here. So one is, in general, the FDA does not like it for people to, if there's an approved drug, to say you can't be on an approved drug during a trial. So there will be, in the future, most things are going to have to deal with DAYBUE, but you're going to have to be a stable dose of DAYBUE. You're not going to be able to start DAYBUE right before you start the gene therapy or anything like that. These two companies are dealing with it very similarly, that either you need to be off of it or stable. And this is the type of thing that it's going to be company by company and how they think about it. And as we learn more and more about DAYBUE and its long-term effect and how much impact it has on a long-term basis, different decisions will be made about how to deal with DAYBUE. Right now, we're at the beginning of DAYBUE and the beginning of gene therapy trials. So rightfully, the companies are being very cautious in not trying to mix things up and confuse things, and try to do a trial as cleanly as possible so they can really know whether the effect that they're seeing with gene therapy is due to the gene therapy and not due to DAYBUE. And that's why originally Neurogene said, no, you can never be on DAYBUE, but they've stepped back from that because the FDA sort of, was like, wait, it's an FDA approved drug. How can you not let someone take it? So but it's challenging because, ideally you'd want people on nothing, but at the same time, if someone's doing well on DAYBUE, as long as they're stable, to add it on, because we know that with DAYBUE, yes, you might get continued slow changes, but you shouldn't get a big change. And so we're hoping for a big change with gene therapy. And so this will evolve, but right now each protocol has their own specific inclusion and exclusion that they use for this, but this is going to evolve with time.
Thank you. Thank you very much. Another common question from a lot of people for today's webinar are about treatment for adults. And I'm wondering if you can comment on the potential improvements and possibility of gene therapy for adults with Rett syndrome.
Yeah, so, if you go back to Dr. Bird's original mouse experiments, he turned the gene back on very late when the mice were mature and were actually, the mice die at some age, and he turned the gene on kind of right before they were going to die, and he saw a rescue of their survival and a rescue of the phenotype in mice. Because of that, scientifically, we are all hopeful that there will be some ability to restore function throughout the life of an individual. Humans are not mice, so how much the mouse is going to tell us about how big of an effect there is in adults, we don't know, and we're going to have to see. But it is something that Tasha started with those over 21, and I would be, if we see big effects, if we see positive effects, and this moves on to be approved, that adults should be able to be treated. How much of an effect we're going to see, and unfortunately, the best model for girls and women with Rett syndrome are girls with women with Rett syndrome. So at some point, we're going to have to just see what happens. But we hope that we will see positive effects, but we'll have to see.
Thank you. So there's nothing necessarily to exclude adults, but degree of impact is to be determined.
Absolutely.
Okay, thank you. I would like if you could just underscore, I know you mentioned this in the slides, but we've had a number of questions about specific types of mutations or variants, and will gene therapy or mRNA treatment work in missense mutations, small deletions, and major exon deletions? Could you just hit on that again, please?
Yeah, so gene therapy, or the current ones that are going on, which is gene replacement, is mutation agnostic, meaning that it doesn't matter what your mutation is, you're just giving back another copy of the gene. So as long as you don't have a functional copy, you're giving back a new copy. So you can have missense mutations, nonsense mutations, small deletions, all gene therapy should be effective. When you get to gene editing or CRISPR, that becomes mutation specific. So there would have to be different therapy for a different guide RNA, a different molecule for each different mutation and each different approach. Though there are, in cells, people have shown that you can actually replace like a whole gene using CRISPR, but that's a little bit far from the clinic, but the technology is there to do that. But for DNA editing, yes, it's going to be very mutation specific. And the same is true with mRNA therapies. So if the therapy is such that it's just going to increase the amount of protein you have, but your protein has a mutation and it's a missense mutation, and as a protein, you don't want to do that. You don't want to bring more of the bad protein in. So that wouldn't be effective. If it's a RNA therapy that causes a read-through, then you need to have a nonsense mutation to do that. So there will be some gene and mutation specific therapies that move forward, but many researchers are thinking about this kind of holistically and how you can help and impact the most people at once. And so this gene replacement is this most straightforward way to do that. But these other approaches hopefully will be able to hit large groups of kids so that you don't have to do individual by individual.
Thank you. I think we have time for maybe three more questions, and then we'll have to conclude for the day. Thank you for taking the question queue. Another question in the queue is, how long until these gene therapy trials could be ready and approved for general population?
So, the process of this is, they start with a few patients, then they wait and do another few patients, and they wait. And so they're both moving through these first steps, and they do it in cohorts, and they're moving through cohort 1 and cohort 2, and there's a little pause in between them. So they're moving forward. So these first couple steps will take a couple years to complete. Once these steps are complete, they're going to look at all of the data that they have and how well the kids have done and how many side effects they've gotten, and decide with the FDA what's next. And hopefully we're going to see huge things in these first 12 patients, which the FDA might say, you know what, just do 6 more. And in a year after those 6, if you see those big, huge things again, we'll give it approval or provisional approval. But it's also possible the FDA says, no, you need to do a phase 3 trial. You need to do a bigger, longer trial, and that could take longer. So it's hard to give you a definitive answer to how long this is going to take, because part of it's going to depend on the results of these first early phase 1, phase 2 trials. And I hope these early phase 1, phase 2 trials will finish enrolling in the next year. And if they finish enrolling the next year, a year after that, we should have enough data to figure out what's going to happen next.
Thank you. I know it's a crystal ball question, but I appreciate your response. Holly, I'd like to get one in for you, please. Going back to the explanation of genetics, we had a question about if there are certain types of genetic changes or variants or mutations or deletions that are more severe than others in the way they present with Rett syndrome without a gene therapy.
Yeah, so we call that in genetic speak genotype-phenotype correlation, meaning sort of does severity or certain symptom type align with the particular genetic change that's present. So we do have some patterns that we know, particularly for the recurrent variants. So the genetic changes that we've identified in large numbers of people with Rett syndrome, we can start to see some patterns where we know some of those variants tend to be more severe, where maybe individuals with that variant tend to have more preserved language, for example, or in general tend to have more preserved ambulation. On the flip side being, we have some genetic variants where we tend to see, we know these tend to be more severe, people tend to be more globally affected. And so we can see that when we look at across people with the same genetic variant. We do know that there's still variability. There are other factors that come into play besides just the genetic variant, and we're still working to understand what all of those factors are. So it's not necessarily like a direct correlation. And again, we don't have a crystal ball to say exactly what symptoms a person will experience based on her genetic change, but at least for some of the recurrent variants, we have some general patterns that we can tend to expect to see. For people who have unique or more rare variants where we don't know as much about, it can sometimes be more challenging to make those types of predictions.
Thank you. So the answer is, in the individual child, could be anything, anywhere on the spectrum, but as an entire population, we do have some trends. And folks can visit our website to read more about that. And we can also discuss that at any of our Ed Days that are coming up. So we hope you can join us. I'd like to close with just 2 more questions, and they will be brief. One is, and I think Dr. Marsh, you may have touched on this, but in terms of deciding inclusion or exclusion criteria for a trial, is that coming from the drug sponsor, from the drug company, or is that coming from the individual PI at one of the individual sites?
Yeah, so that comes from the drug company, from the sponsor. They're the ones who, with the FDA and with talking to all the physicians and researchers out there, come up with the inclusion and exclusion criteria for any given trial. But it's ultimately the decision of the sponsor of the trial to make those determinations.
Great, thank you. And we have a lot of participants today who've joined us from the MECP2 duplication community, and they're asking if any of these Rett syndrome trials are relevant for MECP2 duplication, or if there's a place that they could go to find out what's going on in their particular disorder, if you have any ideas that you could share.
Sure. So yes, there are therapeutic trials about to start in MECP2 duplication. It is actually an ASO trial. The sponsor for that is Ionis Pharmaceuticals, so you can go to their website because I think they have information on their website. And there was also a MECP2 duplication family conference this fall, which I believe that the talks from that, including the talks from Ionis, which talks about their therapy, and a few different other therapeutic approaches that people are moving forward, are all available for access. And we can find out how to get access to those for the community and make that available for people.
Thank you so much. There's, as you said, there's so much optimism in so many disorders, rare and otherwise, and we're very, very excited for this new age, for Rett syndrome and the new potential. And thank you for breaking it down. We will love to have you back again as things evolve and progress and continue, and we expect nothing but great progress as we move forward. So thank you very much. I'd like to remind everyone that the session will be posted on our YouTube channel, so you can go back and digest it again and again and again. And I know that I learn something new every time that I spend time with you guys. So thanks so much for joining us. I want to appreciate all of your attentive commitment to Rett syndrome and your dedication for helping us live better today with Rett syndrome in your clinical work, as well as your participation in research. It's just, you give us all hope and inspiration to do the necessary hard work to live with our loved ones today and see them through all of the changes that the syndrome brings their way. Thanks for your time today. We know you're super busy, so get back to your clinic and your lab, and we wish you such continued success in your efforts. And for explaining this very difficult topic for us. Keep working at it. We're excited for everything that's to come. To our attendees, we hope you found value in today's session and that the knowledge you learned will empower you in ways great and small in your path forward. As you go about the daily care of your child, we want you to take hope that the International Rett Syndrome Foundation is working diligently for better treatments and interventions that are safe and effective in as many individuals with Rett syndrome as possible, young and old, male and female, and across all the different variations that we talked about today. The days may be long, but I know that we're all up to the task when we're empowered with factual guidance and information, and you can count on us to continue to bring sessions like today's your way. And my request to all, but especially to our families, is that you continue to try your best to understand how to advocate for our mostly nonverbal children and to remain optimistic and replenished in your lives, because their success hinges on you. Please take the information learned today, apply it, and share it with others. Believe in them, and we will all be part of something really special. Until we speak again, be well and know that we welcome your feedback on what you need from our IRSF RettEd programming to best empower your extraordinary efforts to raise your incredible loved one with Rett syndrome. Thank you, and this concludes today's webinar. Take care, everybody.